just the few slides I'm going to be sharing with you will give you a little bit of background of what I do. I'm in crop production, and there are many areas of crop production, and some of the component of crop production system are here listed. So we're looking at cropping system, water management, fertility management, and pest and disease management. And I come under the pest and disease management as a plant pathologist. And my research is focused on soil-borne diseases, and we are mandated at looking at three areas of soil-borne disease. First is the etiology. So we basically always doing some survey, identifying new diseases that are on a crop production system, or doing survey to see the changes in existing diseases that we have. And the second area is epidemiology, which is focused on looking at variables that are affecting diseases, so and the agent that are causing those diseases. And the third component is management, and I'm pretty much focused on so-called biorational management of diseases. So in essence, leaning more non-chemical type of management. And we cover a lot of crop from annual to perennial, including chili, all the way to fruit tree and nut tree like pecan and peaches. And one of the fundamental aspects of soil-borne diseases is that the reason they come to elicit the type of damage they're doing come from three functions. They actually affect root, stem, and vascular system of the plant that they're affecting. So when you have dysfunction in any of these three organ, root, stem, or vascular system, then you get wilting, and this is where you're getting a lot of the symptoms that are coming from soil-borne pathogens. Our philosophy of crop management, which can be extended to, I don't know, all of the area in which you're all working in, but this can be extended to beyond crop, to be looking at the ecosystem and by and large. So we're looking at rangeland, pastoral land, but the philosophy we have here is basically captured in this diagram here. So if you look at in the center here, what I have in mind is that in any production system, we have two competing set of variables that are existing in this environment. We have a so-called biotic and abiotic stresses, and then we have so-called biotic and abiotic stimulant. So in this philosophical approach, we think that plants are responding to signals in the production environment. And in order to be able to increase crop performance, one has to be able to manage these signals in the production environment in such a way that you tilt the balance towards the stimulant so that you can reduce the impact of stresses. And you can do that by utilizing several input to be able to manage the signal. So this is really the centrality of my philosophy of crop management. As I said, this can be extended to rangeland, to pastoral land, because at the end of the day, if you're not managing crop, if you are managing, let's say, animal production or any type of rangeland production system you have, your goal is to increase performance. And in that sense, you need to be able to manage the signals in the system. So when we're looking at production system, production environment, I have listed few of them here. This is not all, but we're looking at variable related to soil, edaphic variable, and variable related to the aerial environment. And then we have a biological component. And this is a bias of mine because I come from the Department of Entomology, Plant Pathology with Science. So we pretty much look at the biological entities in terms of pests and pathogen. But there are other entities there that may not be pests or pathogen in the production environment. So as I mentioned in the graph in the previous slide, here are a few of the input that can be utilized to somehow manage this production environment with a goal to increase production or crop production. So one of the input that I'm utilizing or researching on in my program are microbial input. So we're focusing on microbial input from two perspectives. First, to be able to impact the interaction between the pathogen and plant with a goal of reducing the activities of a pathogen on the plant. And the second part is actually managing this interaction in a way to boost the plant performance. So those are the two components through which we are utilizing this microbial input. So when we looking at interaction that boosts plant performance, we have two components. The first one is basically targeting what we call induced systemic resistance in plant. So we're hoping that the interaction, if we have any microbial input, and when they're applied in the plant system, they will help the plant enhance the resistance to the pathogens. And the second component of this are mycorrhizae, which is the topic of what I've been asked to talk on. So in the next few slides, I'm going to go into the general biology of mycorrhizae. I'm going to try to identify what they are, where they come from, how do they make a living, and how do they help plant cope with extreme environmental conditions. Just to give a perspective, mycorrhizae are fungi. They are so-called true fungi. And the fungal statistic that we have available right now put an estimate of about 1,500,000. The figures here are wrong, so you have one more digit than it should be. About 1,500,000 fungi that have been estimated to be on our planet Earth, but only 5 to 7% have been described. So you can see how much is there to be still discovered about fungi. The fungi are classified in this group of eukaryotic organism. So this is a dendrogram giving you overall classification of many of the living organisms we know about. So you have a prokaryote right here, which include the true bacteria and the archaebacteria. And then within the eukaryotic organism, you have several groups of organisms, including the plant, the animal, and so-called the eumycota, which are the true fungi. So as you can see, the true fungi here tend to be basically closely related to the animal, more so than to plant. So here are some few photos of true fungi, and I show this because most people, when go hiking or outdoor, are very familiar with fungi from this perspective. And here are some example of those. And here's another example of true fungi that either encounter in store or in the wood or in a hiking spot that people frequently go on. And here's some example of true fungi. You can see this particular here fungi or mushroom really growing in very drastic environment. This Podaxia species here, which emerged from a sand dune. And over here, another mushroom type called Pysolitis. As you can see around the surrounding, you can see all of the gravel and the dry soil that it seemed to be emerging from. And here are some other photograph of a mushroom uh, that you might have encountered depending on where you're at. So all of these to illustrate what fungi are. One of the challenges I usually encounter is explaining to the general public what is a fungus. So a way for me to be able to explain to them is actually bringing the notion of mushroom, which most people know that either they purchasing them from grocery for gastronomic reason, or when they're out in the wood hiking, they have seen a lot of these organisms. So true fungi are characteristically interesting characteristic is so-called hyphae, which are the building block of true fungi, and they tend to develop this network that you see right here on the right side of the slide. And one of the trademark is that in the cell wall of this hyphae, we have compound like chitin and glucan. Although chitin and glucan are not necessarily only limited to fungi. You can have crustaceous organism like mushroom, shrimp, or crab that do have this in the cell wall also. So they have diverse morphology, but basically they have four growth form, one of which is right here what we call mycelial, and then you have a yeast type. You all heard about yeast. These are part of a true fungi. And the yeast type has two growth form, either the budding form or what we call the fission form. And then the fourth type of growth form is the chytridiaceous, which are well adapted to aquatic environment, which is not the topic today. But anyway, just to give you an idea of the kind of form, growth form that you find behind this mushroom or any other fungi that we'll discuss later. So here is micrograph of mycelium. As indicated, you see a network of hyphae. Okay. And here is a photograph of a network of mycelium coming from a peanut plant that had been infected by this particular fungal pathogen. So you can see the well elaborated net-like of this filament coming from the infected plant. So all of these mycelium, hyphae, mycelium get together, and then we get more different morphology, different fungi, and some of us may have pigment or no pigment. Here is, for instance, a true fungus in a plate, and you can still see your mycelium and impregnated with this pigment here. Now, having defined true fungi, mycorrhizae are typically used. The term is mycorrhizal fungi. There are true fungi that are associated with plant root. And the terminology that you're going to encounter is either mycorrhizae or mycorrhiza, or in singular term. So you have mycorrhiza, or if objective, we're using mycorrhizal. So the headquarter is root. So here's a diagram of a developing root, just to refresh you on the different tissue and the root system. In this transversal cutting here, we see as we move from the epidermal cell towards the center of the root and passing different type of tissue. And mycorrhizal will tend to be associated with a cortical tissue. So we're talking about anywhere between the cortex, between the epidermis here, and within all of this cortical tissue. So there are two basic types of mycorrhizae, the so-called endomycorrhizae and ectomycorrhizae. So this diagram here, in this diagram, I'm going to try to illustrate the differences between the two. So on the left side here, we have a so-called endomycorrhizae, and they are typified by several structures that they form, two of which are the so-called arbuscules. I don't know if you see the head of my arrow over here. So the arbuscule over here, they look like trees, like arbors, that occupy the inner cells, the inner part of the cells. In addition to the arbuscules, they also produce the so-called vesicles. And those two tend to be associated within the plant tissues. And outside the tissue, they tend to produce the structure that we call chlamydospores, which are structures that enable this mycorrhizal to be able to survive in soil in case there is no plant tissue around it to survive. So on the other hand, the other side of it, you have ectomycorrhizae. So notice here, it has two main structures, the so-called the fungal mantles. So these are your mycelium that basically coating the outside of the root tissue, and some filament of mycelium are able to penetrate the tissue of a root and forming a network of mycelium, what we call heartic, as you can see over here, heartic net. So those are the two basic types. And within these two basic types, there are some fragmentation that I'm going to later talk about. So here it is in this table. So recall the endomycorrhizae and the ectomycorrhizae. So the main difference has to do with the type of plant that might be associated with. We have, for instance, endomycorrhizae that are strictly found in orchids. Meanwhile, the so-called arbuscular mycorrhizae tend to be ubiquitous and broad host range across many plant families. So as I indicated, the two main types are endomycorrhizae and ectomycorrhizae. And if you look at the ectomycorrhizae, you have a strictly ectomycorrhizal fungi, and the so-called ectendomycorrhizae tend to have the life of ectomycorrhizae and a little bit of a life of the endomycorrhizae. So continuing on the type of structure of the form, as you can see here, on terms of the endomycorrhizae, they tend to form the so-called arbuscules. So you recall the vesicle that we talked about. And if you compare these two diagrams over here, we have a so-called autumn type and Paris type. So notice over here that we tend to have this mycelium growth that tend to be intercellular, with the exception that they're forming the arbuscule of the vesicle within the cell, whereas in the Paris type, you can see that they're mostly intracellular and they tend to form these coils within the cell of a host plant. So to give you a perspective here, this is a micrograph from under the microscope of what the arbuscule look like. As indicated, look at it, it does look like a tree. And over here, you have a sketch of that. So just to give you a perspective as to what extent the arbuscule network can be formed within the cell of a host plant. So here's a micrograph in which you can see vesicles. So these are trademark again of the endomycorrhizal fungi beside the arbuscules. And in terms of morphology, the endomycorrhizal fungi have a diverse group of fungi, and they can be distinguished by the type of spore that they produce. And you can see here a few of those that have been depicted in this slide. And a lot of this description can only be done under the microscope, unlike the typical mushroom where we can look at the structure naked eyes and be able to go through the characteristic distinction among the mushroom. Over here, we do need the microscope to be able to separate the different genera of endomycorrhizal fungi. So these are some of the main genera of the endomycorrhizal fungi. One of the most common one is the so-called glomus over here. So these are some micrograph illustrating to you some species of glomus. They tend to be subspherical, really with a gold color, and some have a really unique color. And all these need to be extracted through soil through a really tedious method before you're able to acquire them and be able to observe them. So on here are a few more genera beside glomus. You have other genera of endomycorrhizae that are depicted here. Again, microscopy is needed to be able to see, to visualize the spores. Now, in terms of the ectomycorrhizae, as I indicated earlier, uh, they tend to cover the outside of the root with a mantle. So the M here tend for that. So this is a cover, mycelium that cover the outside of the root, and they tend to penetrate also the cortical tissue of the root, which is the hearted neck. So this is a transversal section just to allow you to see. You can see over here all the cortical tissue that have not been colonized that tend to be translucent and clear over here. So here are a few examples of ectomycorrhizal fungi. Here is a seedling of a species of Pinus, and you can see here the extensive network of mycelium that have colonized the root of a seedling. And over here, you have ectomycorrhizal fungi of a mushroom part of a particular mycorrhizal fungi, and showing you how it is branching down into this particular root system and putting in all of the mycelium into that host that is colonized. So many of the roots that are colonized by ectomycorrhizal tend to have what I call some disfigured aspect of the root. So you do see the root structure of it, but they tend to be thicker because of all the presence of the fungal mantle and the heavy colonization that they have undergone through with these ectomycorrhizal fungi. So you can't really miss any of this. If you were to uproot the root, you can clearly see compared to a non-colonized root. So more example here of ectomycorrhizal fungi. As I indicated, they're easy to identify because they do form some kind of mushroom. And as you go through here, you can see we are two genera of 4 different species of ectomycorrhizae, and this can be used to differentiate these different species. Here's another ectomycorrhizae that we call Physolystus tinctorius, very common and is ubiquitous within New Mexico, across the United States, and outside the U.S. And they tend to be associated with a lot of trees. For instance, we find it in a lot of pecan orchard in these areas. And they tend to surge out of the ground, even if, especially in an area that's kind of dry. So it begs the question whether this type of mycorrhizae could be used in drought mitigation. So the beneficiaries of mycorrhizae are about 2/3 of terrestrial plants are known to be associated with mycorrhizae, or the other way, mycorrhizae are associated with about 2/3 of terrestrial plant, of which 80 to 95% are vascular plant. And you have another 3 to 5% of seed plant that are associated with ectomycorrhizal fungi. So what do we know about mycorrhizae that been really attracted about them. So they know to actually play a role, to have function in three areas. First, in terms of plant nutrition and mitigation of environmental stresses, including drought, and mitigation of biological stresses like pathogen and pest. So if you recall again the different type mycorrhizal that I indicated to you earlier, so we have the endomycorrhizal, so most of this, and also the ectomycorrhizae here. So this slide is showing you the typical host plant that they're associated with. And what I really wanted to point in this slide is the type of activity they fulfill in terms of plant nutrition. Chief among this is really phosphorus uptake. Most of the mycorrhizal that have been described primarily known to help plant uptake phosphorus in many of those difficult environments in which plant live. And in return, the plant do supply those mycorrhizal carbon source that are coming from the photosynthetic compound that been secreted by the plant. Another area where mycorrhizal play really have benefit have to do with so-called stabilization of soil aggregate. So mycorrhizal are known to form these glycoprotein known as glomalin, and this help actually enhance soil structures by forming aggregate. And in addition, they help in root exudation, and also that together with the glomalin help actually increase the abundance of soil bacteria. So I'm going to look at briefly some example of benefit to plant nutrition. So here's some work early on several years ago by Graham and Mensch looking at the formation of arbuscular mycorrhizal fungi and condition of where you have no phosphorus and phosphorus applied. So here you can see basically when you have the arbuscular mycorrhizal, they do well when you don't have high fertilization, and in terms of forming the structure that we know to be trademark of these fungi, vesicle, arbuscule. But when you have high fertility conditions, they tend not to produce all of those structures. So over here, we're looking at the same kind of study, the level of phosphorus in dry root. So what this is showing you here is that when you have no fertilization, here's the level of phosphorus in dry root. When you have fertilization, here the level of phosphorus in the non-mycorrhizal fungi. And one thing to notice is that when you have no phosphate apply, the arbuscular mycorrhizal fungi actually help increase the level of phosphate in the dry root. So one of the areas that I have been working on is actually relating mycorrhizal inoculation to disease level in different crops. So this study here, we're looking at 3 different species of glomus, work that was done by Garmendia in Spain. So what is done here, this is in pepper, infected with a vascular pathogen called verticillium. So what I want you to notice here is, let me go back to this, is the disease level. So one thing you notice here is what the green here is no mycorrhizal, and the purple is mycorrhizal. So you can see when you have no mycorrhizal, the disease level increase. Okay. Whereas when you have mycorrhizal present, the disease level is basically decreased relative to a non-mycorrhizal plant. So in another study, we looked at several different species of endomycorrhizal fungi, And this was based on work looking at a soil-borne pathogen called Rhizoctonia in onion. So we're looking at plant height and root length here under different conditions. So one thing to notice here, here's the pathogen, okay? Here's the pathogen activity on the plant height, the yellow, or on the root length, the red. So you can see how much these variables are reduced in the presence of a pathogen alone. And now here is the non-inoculated control, meaning this is the height and root length when you don't have a pathogen. But notice here, when you have the mycorrhizal added, it improves the height and the root length in this onion plant. And even in the presence of a pathogen, is actually improving on those biometric variable. So the same study here is looking at other variable like shoot dry weight, root dry weight, and total dry weight. And again, you want to compare over here the pathogen alone versus when you have a mycorrhizal organism apply. As you can see, there is a greater improvement on the biomass in the presence of a mycorrhizal inoculation. So in my own work here, looking at some soil-borne disease pathogen, so you can see here, wherever we have mycorrhizal apply, the yellow, we have reduced disease incidence compared relative to when we don't have mycorrhizal fungi. So similar thing here, we looked at fungicide in combination with mycorrhizal fungi. One thing you can see here, disease incidence is actually lower when you have a combination of a fungicide and a mycorrhizal fungi. Now, I'm going to move here to talk a little bit briefly in terms of benefit of mycorrhizal in the presence of environmental stress, particularly water stress. So water dynamic include identifying sources of water and the amount of water that might be in the system that you are dealing with. So water sources are here either from precipitation, groundwater, or surface water. And important component of this is the amount or frequency or application of this precipitation and the quality of the water that is being received in the system. And one has to consider the spectrum that go from excessive amount of water to actually lower level water, meaning like drought. So we're fluctuating between flooding and drought condition in any environment, depending on the availability of water in that system. So here are a summary of what I could find from a colleague plant physiologist in terms of indicator of drought tolerance. And these are a few of the listed one. And it goes from not only improving water uptake during those stressful environment, but increasing mineral nutrition, all the way down to actual increasing the induction of plant hormone that are actually important in mediating the response of plant to different stresses, including drought. So the best way to actually summarize the mechanism of drought resistance or tolerance by mycorrhizal fungi is this graph that basically was by Sebastian and colleague. So on the left side over here, you have few of the variables that are being targeted in terms of what we want to see in terms of tolerance, under tolerance, drought tolerance. So what we have is we want to be able to see increased water availability, and also we want to be able to see what we call a reactive oxygen species prevention during drought. So all of these physiological variables that we can measure over here are increased stomatal conductance, And we're also looking at carbon dioxide fixation, the efficiency of this. And one thing also that is important of osmotic adjustment during drought stress. So what the graph is showing you is how certain of these variable actually lead to increase of water and then increases stomatal conductance. And all of these work together to bring recovery in the plant to be able to recover from the drought stress and resume growth. So in this graph here, which was some study done on lettuce by Rio Lozano and Asgon. So we're looking at here nitrate reductase activity. This is an important component that is used also for measuring stress activity, activity of plant and the stress environment. So we have two environmental conditions here of well water lettuce versus drought stress lettuce. So on the left side over here, you have four different species of glomus. So recall glomus is one of the major genera of the endomycorrhizal fungi. So we are looking at total nitrogen reductase activity versus specific nitrogen activity. So total nitrogen activity is all of the sources of nitrogen reductase. However, here we're looking at specific nitrogen reductase activity. So if we look at the yellow, the total nitrogen reductase activity, look at over here the control and well water, you can see the level of that is as good as under phosphate fertilization. But if you see over here under those three glomus species, notice how the nitrate reductase activities increase. Now, if we flip and go to the other side here where we have dry stress, notice in general that the activity of a nitrate reductase is actually reduced due to the drought stress. But one thing to take away from this graph here is that relative to the control over here of a phosphate fertilized plant, when we have glomus presence, when we have plants that have been treated with the endomycorrhizal, you can still see a sustained activity of a nitrate reductase. So in essence, what we acknowledge from this graph is that drought stress will reduce the activity of nitrate reductase, but if you have endomycorrhizal present, they will be able to mitigate the extent, the severity of a reduction by to a great extent. So with that in mind, the question becomes, how do we harness mycorrhizal fungi for agro-ecosystem health? And I use agro-system health in general term, as not only in terms of like pathogen and pest, but in terms of productivity too. So in here, I have in this graph what they call mycorrhizal dependency, meaning this chart allow you to go through dichotomy to actually determine whether or not mycorrhizal are needed in any system. So I understand many of you on this platform are interested in drought. And one thing I would like to discuss with you, hopefully we have time to do that, is that there are two scenarios that could happen, is that to be able to survey drought stress environment to see where we see most of the tolerance taking place and be able to survey whether or not this is associated with some kind of indigenous mycorrhizal fungi. That's one aspect. The other aspect would be whether or not the situation can be mitigated by actually introducing mycorrhizal fungi that have been known to be associated with stress mitigation in other areas. So this graph over here basically is simply guiding how you're going to be able to determine whether or not your indigenous mycorrhizal inoculum or fungi are actually sufficient, or if you might need to actually increase their activity by enhancing those through some kind of input or augmenting their activity by actually introducing some other type of mycorrhizal fungi. So in that sense, the next slide I have here for you, I'm sharing with you, this is looking at mycorrhizal level in rotational crop, free rotational crop here, maize, small grain, and peppers. So, and we are looking at 3 species of glomus. So one thing you're looking at here, what we're measuring is the amount of spore per 50 centimeter cube of soil. So one thing to notice is that the different species are recovered at different level under those 3 different rotational crop. And most uniquely important here is that for the grain, the grassy crop like maize or corn or small grain, we see much more higher recovery of at least several of all of these 3 species of glomus relative to the pepper system. So, which mean in terms of actually enhancing the proliferation of mycorrhizal in any system, one would have to target specific crops that are known to be able to actually enhance their population. So in this case, a lot of small grain tend to be better than like over here with cotyledon, with broadleaf crop like peppers. Now I'm gonna go now talk about mycorrhizal-based commercial product, as I was indicated earlier. So if one is looking into utilizing mycorrhizae for mediating extreme environmental condition in any system you have option 1, encouraging the proliferation of indigenous mycorrhizal fungi, or utilizing commercial-based mycorrhizal product. So the question we ask is, do they work? So there are several products out there in the market, and here are a few of them that I'm showing you on this slide. So again, this show you here some product only go as far by only declaring that they have endomycorrhizal fungi up to mycorrhizae, but they don't tell you the type of endomycorrhizal fungi or ectomycorrhizal fungi it contains. Some have do a better job by actually listing the particular endomycorrhizal fungi they have or the endomycorrhizal fungi they have and the amount of a propagule, or let's say the concentration of each one of us. So here is some work that was done many years ago by a group looking at 12 different type of commercial mycorrhizal product, and most of these contain the so-called endomycorrhizal fungi. And it tells you the number of species that some of them may have, and from 4 to about 10. And they may have other organisms, including ectomycorrhizae, and whether or not they contain fertilizer or not. So one thing here that is important, the reason I'm bringing this to your attention, is that those 12 products on this side here, these are the claimed species that they contain. But the author of this research, once we're done, these are the species that were detected. So as you can see, for the most part, there is a net demarcation difference between the claimed species and the species detected. And they also tested this product in terms of colonization of root, across in soil, as in this particular case here, to see to what extent they do see root colonization. And they also did it in sand, which doesn't have any other nutrients. And you can see over here that only few products were able to demonstrate that out of the 12 products they've seen, but only few of the products show some activity in terms of mycorrhizal colonization of root. Now, this brings me to the last part of a presentation, which is if you are going to be utilizing commercial product containing mycorrhizae, you need to do your own tests. And this is showing a few of things that we do in the lab. Basically, we choose at least different rate and do some treatment where we do field testing where we have treated and non-treated. This can be done under a controlled environment. So you have an idea whether or not the product that you are going or planning to utilize basically has the claim property that has been prescribed. So I would like to finish up here and take any question that you may have.
Thanks for your presentation. Really appreciate that. I do have one or two questions here. The first one, kind of what you were saying with your inoculants. What is the best method for applying those inoculants? And I'm sorry if I missed that somewhere.
Um, Most of the product call for they have free application either to the seed. Okay, so you can treat the seed like what I do with grower here. We do a seed treatment right in the planter box or
Mhm.
at planting.
Yeah,
We also do the application of it.
right.
So either by directly on the seed or side dressing, but for the most part, a lot of these mycorrhizal fungi call for seed treatment.
Okay.
So that's
Okay.
the
And
best.
that's good to know because I'm doing a field test with some endomycorrhizae inoculant, and it's, we're on a couple acres here, and so we're gonna
Oh,
plant in a planter box we're gonna plant, with that, and so I just want to make sure, if there's maybe a better method for applying aside from just directly onto the seed, that would be great. Okay,
so what
thank
are you
you.
planting?
We're just gonna be doing some pinto beans.
Pinto beans. Okay. Yeah, I think pinto beans are large seed and should be good, amenable to getting good coverage
Okay.
of a product. Yes. So the other thing too, um, I don't know when you're gonna plant. Do you know when you're gonna plant?
Probably not till May.
Oh, okay. I was gonna say, did you test? Do you know how many, what is in your product? Is it one species, two species, the
Yeah,
product you're using?
it's called bioorganics, and it's a whole bunch of glomulus species, but I've not actually done any kind of lab testing to know what's really in it or not. So,
Okay, all right. Well, yeah, I was gonna say that um, typically if you have enough time, although it's hard to do sometime, is to actually see if you can, just like the last slide I showed you here.
mhm.
Let me see if I can share that again.
Still sharing. We can still see it.
Oh, it's still sharing?
Yeah.
Oh, okay. Can you see that one?
Yes.
Okay. So what we did is we use this was wheat and onions, but we just treated this side and uh, we wanted to see if really there was any difference between to be sure that the product was really working. So we did this growth chamber study just to be sure that it's working before we go to the field. So I was gonna say maybe before May. I know pinto bean grow really fast. Uh, you could quickly do something like this if you have access to growth chamber. Do treated and non-treated and see how it's growing. You
Yeah.
know,
Yeah.
just to get an idea what is actually working.
Thank you so much. I appreciate that.
Christy, I see you have a question in the chat. You want to?
Sure. I was just curious if you had any experience with like the Rodale Institute piloted a way to culture your own arbuscular mycorrhiza on site. Um, do you have any experience with that?
No, no, I don't do my own, but I only test it for people who really, for instance, on that slide, I show you that most of the way commercial product or company really raise with mycorrhizae, they use some kind of grassy crop, so wheat,
Hmm.
oat, and all of these because they're easier to quantify and easier to grind, but I don't have my own production on the site.
Okay. Thank you.
Yeah.
So I want to ask. I noticed in one of the studies, it was citing Glomus deserticola, which I assume is from a desert environment, and it's just sparked for me the question of whether the mycorrhiza that are found in arid environments are maybe particularly effective for mitigating drought stress, or if there's any sort of pattern there.
Don't think I'm aware of anybody who's done systematic comparison of, say, because a lot of these species could be found in many other areas. You could find glomus glomus, let's say, mostly in Iowa or say Maryland. So it will be nice to see if somebody can do study to show desert species isolated in arid soil versus soil under heavy precipitation area, if they basically different in terms of a colonization ability. And it hasn't been done. In fact, when Guillermo mentioned this to me, and the reason I went this way, because I was kind of thinking you guys gonna take me on this challenge aspect of how do we mitigate drought stress, say in arid area or arid area within area that we know are not arid. For instance, a desert area in Washington State, okay, versus desert area in New Mexico. Okay, so how do you mitigate on in this area? They're planting different crops. So what would be the strategy for utilizing mycorrhizae? So to your question, I don't, that study has not been done as far as I know. All I know is that people in Spain, they've done a lot of work doing survey actually of desert environment, trying to look at different vegetation, see the type of mycorrhizae that they're encountering. And the conclusion is that the predominant vegetation tend to actually have more of these different species of the mycorrhizae than the non-predominant species. But I would think, to your point, a research might be needed to do that. But we have to ask the question then, if they're only limited to the environment they are in, the question will be, if you have, like I said, in the extreme, something coming from a high soil moisture environment, will they withstand in the area of New Mexico, Arizona, and other places?
Guillermo, go ahead.
Hey, Dr. Sanogo, thanks for your presentation and I guess finally making it to give us this talk on this topic. Since you have this slide up, something that I was thinking as you were going through it is, besides fertilization, is there something you have seen that like traditional farmers do to their fields that actually helps the indigenous mycorrhiza community? Or maybe have you seen something that negatively affects the indigenous mycorrhizal community?
Yes. So by and large, high fertilization actually will inhibit mycorrhizal community. So it's basically counterproductive. And a lot of growers tend to actually put a lot of manure in the field. And so to a point where the native mycorrhizal fungi have not been given a chance really to develop, if I may use that word, because high fertility field. Now, many of them, and this is from the perspective of disease control, are trying to use what we call probiotics, which include mycorrhizae, so agricultural probiotics. So this is the trend of most of them. And one thing I have recommended to them to do is that they will plant a fall cover crop and apply the probiotic in it with the hope that the probiotic will be actually fixated in the crop, in the root, so that they have high level of the microorganism come into the spring. Okay, so I hope this is answering your question here. They're all interested in mycorrhizal work, but I think for the longest they've all been doing the wrong thing because they do put the mycorrhizal product, but they also continue to fertilize it heavily. So as you have seen in one of the slides, when you have heavy fertilization, it basically makes the mycorrhizae, I call it lazy. Okay, so it basically inhibit mycorrhizal activities. So at this moment, my recommendation has been working many growers here is applying in the fall, in fall crop, so that there is something at least residual level of a mycorrhizae come in the spring. In fact, I'm even recommending not one season, perhaps to go 1 or 2 seasons if they're able, because no one has done study to show, like what I recommended is not really that I have done systematic study to show that. Is one cropping in fall enough to actually bring the mycorrhizal level to a point that it might be satisfactory to the grower? And in this case, their goal is disease management. Okay. And we haven't even looked at other aspect, which is drought, or let's say irrigation. Okay, for instance, most grower could probably combine two things, not only looking at disease control, but they could also looking at water economy if they decided to do like deficit irrigation. Okay, deficit irrigation is not drought stress, but somehow is going towards there. So it would be nice to have a study that combine mycorrhizae under those conditions so that at least for your network, okay, for your network on drought, so one could say midway could be deficit irrigation. Could deficit irrigation be actually strengthened by mycorrhizal application? So that could be something that could be done. I haven't done that research, but these are ideas that could be explored.
Can I just follow up with that and ask what you would use as the probiotic?
Well, there are several species of fungal-based probiotic. One of the main fungal-based probiotic are so-called trichoderma species. There are several of those on the market. And then there are several bacteria, a lot of bacillus bacteria that most growers are using. Now, what has not been shown is if you combine trichoderma-based probiotic, bacteria-based bacillus probiotic, or even the mycorrhizae probiotics, I call it, with some kind of irrigation, where will it lead you to? So these are all open questions that can be researched.
Really interesting. I'll add one more. So, at one point you discussed, inoculation versus management that encourages the indigenous mycorrhizae. And I'm wondering, especially for like a rangeland setting, some of our ag systems in the Southwest, really extensive rangelands, like, do we know anything about what practices might encourage mycorrhiza that could mitigate these stresses in that kind of setting?
No, actually, thank you guys for inviting me. As I came to this, it opened up a lot of, what do we call it, Pandora box to me, because um, I know the USDA Jornada Center has done a lot of work in terms of looking at the native grasses and all of this and how and I know Mary Lucero and some other scientists have looked at endophytic fungi, okay, which is another group of fungi different from a mycorrhizae, to see how they actually help plant withstand stress. So to your question, I am not aware of any study that have looked at mycorrhizae in terms of stress resiliences in our system. I would like to see that a survey of those grasses to see wherever we have a good stand of grasses, for instance, if that can be attributed to some kind of mycorrhizae. But again, this is kind of foresighted, that's kind of bias in a way. We're cutting everything to mycorrhizae, but we also have to be aware of the possibilities that it could be involving bacteria. So we might probably have bacteria, soil bacteria, that are able to withstand drought, high heat, that could be at play. So really, although this is emphasizing mycorrhizae, my presentation didn't bring all of this into it, but we also have to see, are mycorrhizae acting alone? Or are they acting in consortium? So that's why a survey, directed survey, will be able to tell us that. So if we find good stand versus not good stand, okay, and be able to look at the whole microbial profile from the endophytic fungi, bacteria, into the root system to look at the mycorrhizae and also the bacteria, then we might be able to have a complete picture. And with that, we might be able to formulate mitigation process, at least in terms of enhancing, okay, the existing microbe, or how do we dispatch this microbial population from the good stand into the poor stand area, assuming, again, there is no other confounding variable like edaphic. So because a bad stand could be also maybe soil related, but purely physical. So you would have to involve multi-layer juxtapositions of data, microbial data, physical data, and then be able to charter a mitigation way forward.
Well, yeah, so many directions to explore. I mean, thank you for a really fascinating presentation. And thanks everybody for joining. Yeah, I guess we'll conclude there unless anybody has a burning final question. And I'll just say stay tuned for our next meeting in May.
Thank you. I appreciate the
Yes.
invitation. So if there is anything outside of this that you want to explore, feel free to contact me. I'll be happy to continue outside of this presentation discussion.
All right. Thank you so much.
All right.
Thank you.
Take care.
Thank you.
Thanks, everyone.